Emily? What the heck? What happened?
I've been sick. I've tried everything. I just can't get rid of it.
Oh, well, here you go. Try this.
What is this?
It's dirt. It's antibiotics.
What?
So you may ask yourself, what is Tiny Earth about? So Tiny Earth is a collaboration of students, professors, and scientists who gather together to find antibiotics. And you may ask yourself, how do we find antibiotics? Well, we get it through soil. That right there is where I got my soil from. And you may ask yourself, why do we need to find antibiotics through soil? Well, in the future, antibiotic resistance is going to be the leading cause of death globally. Around 4.9 million people are dying due to antibiotic resistance. And that's insane! Alrighty then, first things first. After we gather our soil, we then are in a lab setting where we measure our soil first to put the data in our Tiny Earth profile, and then we start the dilution process. Here are our materials, what we use to dilute our soil and then place it on the plates. So now we have our prepped plates. We also have our tubes filled with saline, micropipettes, and tips, and we have Matthew labeling. Then we have Matthew grab the original solution and put it in the test tube filled with saline. As you can see, he is putting it on the third test tube. And you can see him go up and down, just so we can mix it up a bit. And then we'll grab that solution and pretty much do the dilution, so that way it gets less and less and less each time. So to spread the sample, we used a glass spreader, dipped it in ethanol and covered it in flame, allowed it to cool for 10 seconds. Then we added 100 microliters of the suspension to the labeled plate and distribute with the sterilized spreader immediately. So I wanted to take a turn, and I grabbed the micropipette, grabbed the tip, and continued on with the third and fourth dilution, so that way I could put it on the plate and use the glass spreader to spread it. And repeated the same process as Matthew did the first time. So this one we wanted to measure the pH of our soil mixed with saline. As we finished up the experiment and the day, Matthew collected all the plates together that we already prepped, and then we put it on the incubator, and we say goodnight to our bacteria as they grow for the next time we come back. So now we'll see what was in our plate. So now we'll start with our 4 diluted plates. On the far left side, we have our first dilution, which looks like there's a lot of stuff going on, and you may be wondering, what is on that? Well, it's a mix of colonies and a mix of bacterial fungus that we'll be talking about soon. The second one will be our dilution 2, which still has a lot of things going on, but we see a tad bit less of it. Then we move on to our dilution 3, moving to the right. We see a lot less of that bacterial fungus and more so of our colonies. And then we'll hit to dilution 4, where we see less of that bacterial fungus and a little bit less of our colonies too. Well, that's called Bacillus mycoides. So Bacillus mycoides is actually a spore-forming bacteria that is widely spread in nature. And as if you can see on the video, you'll see a bunch of little squiggly lines and swirls on the plates. Yep, that would be Bacillus mycoides. And that's something that we want to stay away from. We consider that the quote-unquote monster because it can contaminate our antibiotic-producing microbes. And that's something that we don't want that to happen.
This is where we hold them! This is where we fight! This is where they die! And these shields, boys! Remember this day, man, for it will be yours for all time!
Spartans! Lay down your weapons!
you saw from the previous video, what we see here is our colonies fighting back the mycoides. Now, how do we know that? Well, if you look at the video closely from what I've inputted, you see that there's a ring, and in that ring is like space in between colonies and the mycoides. So that means that our colonies are actually fighting back, which is awesome. But wait, we gotta ask ourselves a question. How do mutations work? So there's actually a beautiful thing in the nature called mutations. Now, so some of our colonies were going through mutations to where they withstood the environment that was filled and contaminated with mycoides. Now, this is a great stepping stone process to where we can see our colonies eventually developing antibiotics. So we might ask ourselves, how do mutations actually work? Well, while doing research, we found that it states mutations result either from errors in the DNA replication or from the damaging effects of mutagens, such as chemicals and radiation, which react with DNA and change the structures of the individual nucleotides. And that means bacteria in a harsh environment will somehow eventually develop a mutation where they can withstand that, which is actually very interesting because that's what we see going on with our colonies. Another great example that you'll see in this video is a giant plate agar made by Harvard. Now, this experiment was done to show the resistance against antibiotics, and we see bacteria growing and we see them developing mutations as well, which why it applies to our research today and why we have Tiny Earth today. Now, this can also be applied to our colonies as well, which they're developing mutations. After talking about mutations, now let's get focused on creating our library. So during this time, my partner and I gathered our chosen fighters to create a library. The purpose of having this library for our bacteria is so that way we collect the growth of that developed bacteria and have it incubated so that way they can grow in a new environment that does not contain mycoides. And throughout this process, we did it using a technique called patching. Let's take a closer look at the library. This is the same fighter bacteria we gathered, and as you can see in the picture, something that seems to be different. If you took a guess as to what it is, you are probably right, and that is the bacteria having different shapes, sizes, slight bit of color, a little bit of transparency, and more. This was very fascinating to see, that these fighter bacteria carry a characteristic within themselves, so that the bacteria can grow without the presence of mycoides. So right here in this video, I have myself being recorded of transferring our bacteria that we have chosen to be our fighter bacteria and move them over to a new agar plate. In this technique of patching, I'm actually using toothpicks to gather my bacteria source and moving it to another plate. Now, these toothpicks are actually autoclaved, meaning that they are sterilized to the point where there's no source of bacteria to where I can contaminate anything. So I'm grabbing my toothpicks from one sterilized side, I grab my bacteria, put it in another plate, and on the left hand side you can see that there is a disposable beaker where I put my toothpicks in. Anabiosis? What's that? Anabiosis is an antagonistic association between two organisms, especially microorganisms, in which one is adversely affected. Now, for this part of the experiment, we are going to put our colonies to the test to see if they will be able to handle against the target bacteria known as the escape pathogens. So, the escape pathogens are contributors to antibiotic resistance, and we will repeat the same steps and techniques as we used in the previous experiments. I don't want to leave you hanging about what escape pathogens are. So, on this picture on the right side, escape is actually an acronym meaning all these pathogens that are right here. So we have E. coli, Staphylococcus, Pseudomonas that contribute again to being antibiotic resistant or growing a mutation of antibiotic resistance. With our station prepped, we have our overnight broth culture that we're going to use for a dilution process, which I have my micropipette here set to 100 microliters, gathering from each pathogen that we're using, which is E. coli, Bacillus, and Staphylococcus. Meanwhile, we have our control right there with the black top that we're trying to match with the saline solution. and you can see there's a bit of a match here. And once we do that, we'll be dipping a cotton swab and swabbing it across the plate. So here's a snippet of me doing a dilution where I grab 100 microliters of the pathogen and I'm mixing it with the saline solution, trying to match the control. Then here's my partner Matthew, who is doing the swabbing. So he grabbed a cotton swab and he dipped it into the saline solution filled with that tiny bit of pathogen, and now he's spreading it across the plate so that way we can patch our library onto that. Now, it may seem like Matthew's doing all the work. I promise you I did it too. I just can't record both of us. So, yes, just know I do my equal share as well. Now, once both of us finished the swabbing across the plates, now it's time for the patching, where we again use an autoclave toothpick grab from our library, and then we transfer it over to the plate covered in the pathogen. And just to let you know, we do label our plates as E, S, and B, and you can tell those are the pathogens. So yes. So this is our result, and on this Staphylococcus plate, we see about 3 of our colonies gaining a mutation against Staphylococcus, which is awesome because that means that those colonies are producing resistance against Staphylococcus. We see the same for Bacillus, where you can see the blue dots and 1 red dot, that there is also a ring or a zone of inhibition. This one, unfortunately, we did not see a ring or zone of inhibition, and we even waited a couple more days to see if the colonies would produce such, but this one did not work. Afterwards, we were given the task to team up together with other group members and combine our successful colonies to once again go through antibiosis. The chart that's shown in the center is all of our group members, including mine, combined of our colonies that we have chosen. After the streaking process and incubation, here are the results that we received. Individually on my end, this is what my plate looks like, as well as the other one. And you can see that there are more circular shapes. I also have pictures of my group members, and this picture is when we let our bacteria incubate for a longer period of time. We also notice a slight color change as well for some of these streaks. Now this is us as a group running the antibiosis again, this time with different pathogens that we were assigned with. Each group had to swab a thin layer of the pathogen that they were assigned, and then we had to spread it across to each other group and patch our colonies. Finally, getting to the end of the video, here's the data from our colonies and showing how their inhibition was shown throughout the lawn of pathogens that were set on the agar plate. Afterwards, we sent this data to Tiny Earth, and now we let them handle the rest. Thank you very much for sticking to this video, and hope you stay happy and healthy.
Well, I hope you enjoyed my Tiny Earth project.